So good morning, very good morning, my dear students. So today we are going to continue with the second part of the biological control of pest and disease. So in the last class, we were discussing about the what is actually meant by the biological control. So what are the importance of the biological control? What are the advantages and disadvantages of the biological control when comparing with the chemical control? So what are the different categories of the biological control agents you can use for the biological control, predators, parasitoids? And today we are going to continue with the pathogens. And what are the broad categories of the biological control methods, right? So the conservation, inundation, and classical biological controls. So this classical biological controls means so you have to introduce any biological control agent where it originated into the somewhere where the pest identified, right? So in this way, in this today's class, we are going to continue with this how pathogens, right? How pathogens are going to use to manage insect pests. Right. Thereafter, we will continue with the weed biological control and the how to manage the plant diseases by using these antagonistic or the biocontrol pathogens. Right. So the insect, the pathogens that are attacking on the insect, we call it as an entomopathogens. Right, entomopathogens. So these entomopathogens mean their target is to eat or kill or used as a host the insects, right? So you can classify these entomopathogens into different categories: viruses, so bacteria, so microsporidins, entomopathogenic fungi, entomopathogenic nematode, and protozoans are there. Some of the protozoans are also there. Some of the mites are also there. Right? Some of the mites are also. These two things are not here. But that's the thing important of the attending of the class to collect the extra points, right? So viruses. So normally there are two types of viruses: polyhedrosis viruses and granulosis viruses. There are the two different categories of viruses. So they are targeting on the we are targeting to manage these insects. We call it as, and these are entomopathogenic viruses, right? Polyhedrosis virus and granulosis virus. And the bacteria, normally we are using, so very popular one is the bacillus, right? Bacillus thuringiensis, bacillus populi, and different type of bacteria are highly using used by the scientists, and it has been commercialized. And microporodin, so this is, I called you the protozoan, some of the protozoan against the grasshoppers, right? Nosema locusti is one of the very important and the famous microsporidins. So sometimes we call microsporidins, we call it as in protozoans, right? Protozoan, Nosema locusti. So especially for the locust control. And entomopathogenic fungi, there are several entomopathogenic fungi has been reported, and in the next slides I will discuss, and the entomopathogenic nematodes too. And some of the predatory mites also we are using and to control, or parasitic mites to control these insects. Right. So these are the ways, right? These are the ways they can cause the diseases. You see, so these entomopathogens, especially the fungals and bacteria, so is how they are attacking first for the accretion of the spores to the host cuticle. Normally, they are spores. So sometimes we call it as if you are taking the bacteria, right? So bacteria is vaginated, so ingested into the body cavity. So while digestion, it will release some of the toxins we call it as an endotoxin. For example, bacteria. I'm explaining bacteria, right? The entomopathogenic bacteria, bacillus, especially the bacillus. So these released the endotoxins are capable to attack the insect gut when this protein coat of the endotoxin abolished. Therefore, so this is designed because normally the insect gut is alkaline pH. So therefore, these bacteria toxins will be released, and the protein coat will be abolished in the alkaline pH conditions. That's how these entomopathogenic bacteria are working. Right? That's how they are non-toxic to the human being and higher animals. Right? So in this way, by using this gene, which genes encoding this endotoxin, nowadays they have modified many crops, GM crops, BT gene inserted crops, mainly maize, cotton, right? Or BT brinjal in India. But the consumer preference are less, and the consumers are claiming that we should not use, or if we are using that, there are several side effects are there due to these genetically modified organisms. But as a scientist, so as an agricultural scientist, you should also know that one. So there are no any side effects by using these BT gene inserted maize or brinjal, all things, right? So if this genetically modified crops, some of the modified crops, they have the drawbacks. Why? If they are changing some of these proteins to target the insect or pest, then these proteins can cause some detrimental effect to the high animals too. But this case, this is purely for toxin, no any other modifications. If it is no other modifications, these are safe to the consumption, right? Safe to the consumption. So that's in this way. This is we call it as a mode of action of these bacteria. Entomopathogenic bacteria mean, so they have that endotoxin. This endotoxin is released when it is digested by these alkaline pHs. So these toxins are penetrated through the midgut because there is no any cuticle in the midgut. Then it finally the bacteria toxins and bacteria multiply, penetrate into the gut and hemosyl, and it can cause septic or sometimes. So this toxin can dangerous and it will kill these organisms. In this way, this BT toxin is working. If you are taking the fungi, entomopathogenic fungi, especially spores, right? The spores, adhesive spores are used to control the insects as an inoculum. Right? The spores adhere to the cuticle and it will produce, as usual, as a disease like initiating, they will produce germ tube and the penetration peck. These penetration peck with penetrated, once it penetrated in mycelia, grow and invade inside the insect body, and it will absorb all digestive content, all the content inside the insect body, and finally the insect will die due to the starvation and also the toxin produced by this fungi, right? So these are some of the entomopathogenic fungi has been reported and popularly used for the many agriculturally important pests. First, one such example is Beauveria bassiana. It's a Beauveria bassiana, we call it as an white muscardine fungi. We call it as a white and green muscardine fungi. So this is mainly used to control the whatever the beetles, especially the coleopterans and the caterpillars, right? Caterpillars. As like the Metarrhizium anisoplia, also the same. Right? Metaprisium anisoplia. Lecanicilium lecani. This is Lecanicilium lecani. You can also control, and Bacillomyces and Lecanicilium you can also use to control the nematode as well, right? So these insect groups and the Nomoria, Hirsutula. Hirsutula we are using especially to control the mites against the coconut mites. Hirsutula thomsoni. So Nomoria reli. This Nomoria reli is used for many pests. See, these insect mainly we are targeting on the aphids, right? Aphids, white flies, mealybugs, and the lepidopterans. Like Bacillomyces and Nomoria, You can use against the thrips and white flies and the aphids. Beauveria, Metarrhizium, you can use against the Lepidopteran and Coleopteran, grubs and adults. Right? Adults. So, and also you can use to treat the you can mix this one with the compost and you can apply to the crops. So whatever the grub inside the soil also, these fungi can attack and kill, right? Attack and kill. So this is a Japanese beetles, right? Japanese beetle, they controlled by using this granulosis virus, right? So granulosis viruses, this is a commercial product, milky spore, right? Milky spore. So the granulosis virus are used to control these Japanese beetle, right? So not the viruses, this is a bacillus. Bacillus populae spores they used to control the Japanese beetle because the Japanese beetle are very big problem in these vineyards. And this is the virus attack, right? This is a virus attack, nucleopolyhedrosis virus attack larva. How its lava's colors has changed into blackish, right? If you are touching that body, so it will like a rotate, it will go inside. It will go inside. If you are applying that one, the entomopathogenic nematodes, so not entomopathogenic viruses, in 1 or 2 days, the larva will die. So before dying, what will do the larva? Larva will climb over the tree, right? Climb over the tree, and from the aerial part, it will hang upside down, right? Upside down. So why? This is due to the viruses behavior. Virus need to spread, no? So as an occlusion bodies, no? Virus need for the spread. They are inducing the larva to hang in upside down. Therefore, to the oral, is bacteria, these viruses can spread to the other, right? Spread to the other. Main target of this nucleopolyhedrosis virus and granulosis virus to manage the lepidopteran, right? Lepidopteran larva. It is very successful in any environmental condition. It can work, but the amount is very less, right? Amount is very less. Amount means the culture and purified cultures are less. Oh, this is polyhedrosis virus, moth larva infected with the polyhedrosis virus, you see. And these top caterpillars, right? So top caterpillars infected with the virus, you see healthy and infected virus. Color will be changed, right? So sometimes this insect will not move. It will become a lethargy. If the bacteria, if the larva is infected with the bacteria, it will vomit, right? It will vomit. So these are the indicators. So green, the green clover grounds attacked with the fungus, you see. So if you are finding anywhere the dead larvas, so it is better to identify whether it is attacked by any pathogens. What you can do is you can take this larva and keep it under the moisture chamber, then you can observe under microscope, you can find it if the fungal attack is there, right? So if the larva color is changing to reddish color, that is reddish brownish color, then it is indication of the entomopathogenic nematode attack. The larval color is changing like a faded color. It is and the mycelium is growing, it's an indication of this entomopathogenic fungal attack. The larva become blackish, is an indication of this viral attack. You can remember easily and you can observe that one, right? This is a fly attacked with the fungus. You see, so this is fungal we call it as an entomophthora. Entomophthora. So mainly we are targeting over the flies. It's naturally available fungus. So even though we can commercially cultivate and spray the fly control, entomophthora. You see all the spores. This is Japanese. Japanese beetle larva has a milky disease of this bacteria. Milky disease. You see it's rotting. Colors has in changing. We call it as a milky disease, mean fade larval become whitey. You see blackish color, it will become white. That's what we call a milky disease. The larva will become a milky color. Right? This is an entomopathogenic nematode attacks. There are two genus of these entomopathogens belongs to two families, Steinanima, Heterorhabdidae. Steinenematidae, Heterorhabdidae, these two families, entomopathogenic nematode. So two genus, Steinanima and Heterorhabdida. Steinanima, Heterorhabdida or Heterorhabdidis. These two genus are highly being used. Why? So these nematodes can kill the insect within 48 hours, and it will multiply in millions in 48 hours. So that's how these entomopathogenic nematodes are also very good biological control agent to manage especially the grubs, right, and lavas. Whatever the lava grubs are in the soil, also you can use these entomopathogenic nematodes. Those entomopathogenic nematodes, what they will do, they have then bacterial associations, right? They will not kill. Nematode alone will not kill. So inside the gut, they have the bacteria. We call it as an Photorhabdus and Xenorhabdus, two bacteria. This bacteria, once the nematode penetrated into the body, how it will penetrate? Right through the natural openings, mouth, oral cavity, and spiracle and anal opening, and in between the scleri they have some delicate membrane. No? Through that nematode will and vomit that bacteria, whatever the bacteria in the gut, no, these Photorhabdus and Xenorhabdus, vomit into the larval hemocoel or larval or grub hemocoel. This bacteria will multiply and kill the insect. Thereafter nematode will eat. You see how it is associated, because the bacteria in the outside environment, it will not work. Therefore, they are living in the nematode guts. But nematode cannot, they doesn't have the ability to kill these insects. So they need the help of bacteria. That is we call it as an both are getting benefit, right? Both are getting. So due to the septicemia, bacteria is multiplying now inside the body. Due to the septic, so in the insects will die. Insects will die. So these are the entomopathogens, how they are attacking and killing the pathogen, the pest. That's what we call as an entomopathogens. So now we are going to move these weed biological control. So weed also the pest. So you in the agriculture cultivations and for that you can use to control the weed. How you are using these biological control to biological control agent to control the insect pests. So weed is a plant. Therefore, whatever the disease causing agents on these weeds, we can use to control the weed. But the problem is we have to thoroughly check. So because you are going to introduce a pathogen, if the weed belongs to same family of the crop, you have to check it whether the target effects are there or not. Therefore, so biological control of weed by using the pathogens are limited. But weed biological control by using the insects having also the same conditions, but there are some of the insects has been identified and it is popular around the world, and we will discuss, right? Weed biological control also there are some advantages and disadvantages, and I already explained that one. So this is a classical biological control process of this introduction of the weed biological control agents. It is there, and I have explained this in the last class too. So arthropods you can use very easily, insects and mites. Right, insects and mites to control these nasty weeds. These are the successful weed species and control agent introduced in different part of the country, right? So especially used to control the different wheat groups, including Parthenium, right? Including Parthenium, and some of them are introduced even in the Sri Lanka to control the cactus species, right? Opuntia stricta and Optunia vulgar. So these are the weed species, and Podia crassicava. These two are the cactus species, and some of these weed species also controlled by introduce some of these weeds, weed biological control agents, so especially insects. So we will see one by one. This is first success in Australia, right? So they use this coleopteran beetle, Chrysomelidae, right? So to control the Alternanthera the phylloxeris. So this is a wheat, Alternanthera, like an what do we call, I don't know about in single names in Ponangani. So Alternanthera sessilis, right? Like that. So some of the plants, this belongs to the same family, but are weeds, right? Problematic broadleaf weeds. So they have used this beetle, we call it as an Agasclis hygrosphylla, right? Agasclis hygrosphylla to control the, this is we call it as an alligator weed. This is called, we call it as an alligator weed. This weed is problematic mainly in the intermediate and wet zones of Sri Lanka. Right? Intermediate wet zones of Sri Lanka also we can use this one, but we have to thoroughly check, right? Thoroughly check before introduction of that. Another one also they used to control Western United States to control the transit ragwort, right? To control the transit ragwort. So the Longitarsus, right? The Longitarsus, this is also beetle, right? Coleoptera to control these weeds, like how do we call for chrysanthemum, right? Chrysanthemum like this. So they used to control that. Another one is there in the USA, same. So to control the Ephobia species, right? So the Ephobia species they use is a very big problem. So they used another beetle we call it as an Apothonaisis, right? So an Apothonai species they used. There are two different categories. Apothonai species also they have used. This is another one. It is very successful even in Sri Lanka. They introduced, right? So Neochitina. So Neochitina weevil is they have introduced to control water hygiene. Water hygiene is very problematic beyond the Vaunia. There is no problem in Venni or in Jaffna because there is no any. In Venni there are a lot of ponds, but these weeds hasn't spread into the Venni area, but it is there in the Vaunia and beyond. We are going there Anuradhapura. So along the Mata, whatever the ponds, you can find this water hygiene. Green color plant with a purplish color flower. This like an mimic to the lotus, right? Lotus. So this for they have introduced that Neochitina species into Sri Lanka. The scrubs and the adults they are getting they are damage. They are making damage, but the success rate is less. But it is continuously available and continuously they are eating. Right? How they are managing by man, human, they are involving to clearing, and nowadays they are making as a compost of these weeds, right? Compost of these weeds. You see, this is a water hygiene, right? This is a water hygiene. Everywhere in Sri Lanka, you can find it beyond the Vonia. So this has all spread. But the advantages of this weed, if you are continuously clearing and making as a compost, it's a good nutrition-rich organic fertilizer. Right? But people are not much interested, right? People are not much interested. So if you are composting, we can use it. And another important thing is you can use this one as a phytoremediation to remove the excess nitrogen or other things from the water. That's how these are growing in the water bodies. Why? So because it's excess nitrogen, because of over application of the water. So leachates all are there in the water bodies. That's how this weed is high. Okay. So different ways you can manage. One of these is Neochitina weevils you can easily use. Right? After introduction of the Neochitina weevil, you see before introduction, after introduction of these Neochitina weevils, they are eating and grabbing and the new green leaf contents. Now you see. So overall you have, it is managed, it's a success. This is not in Sri Lanka. This is some part of the US. Right? Another one is there. You all know that one's Salvinia wheat. So in Second World War time, so many the US and the British people they use Salvinia to cover the water bodies, right? So in the war time, if the pilot can see that, because at the time there was no proper development of the very zooming cameras. So what they have done is they have introduced these weeds all over the water bodies. It look the pilot from the area, the aerial part of the plane. If he is finding, and it will like a that with the grassy land. What he will do? He will land the flights. Finally, flight emerge and it will collapse. For that tactics, they introduce that Salvinia. So because in 1 to 2 days, it will cover the full water bodies. It look like a green if you are in the tall places. If you are finding that, it's like a green places. You cannot find the water. So now the people are just because it is covering. If it is covering the whole surface body, the water cannot get enough oxygen. The plant underside of the water, it cannot photosynthesize. So it's a problematic. Nowadays, what they are doing is they have introduced another weevil we call it as a Cryptobagus salvia. It is also introduced in Sri Lanka. It is very successful. Nowadays, you cannot find much salvinia in all over the water bodies, right? It has been controlled, right? Controlled. So Cryptobagus salvinia, right? Salvinia mollusta is a wheat name. To control the Salvinia mollusta, they introduced a weevil, we call it as a Cryptobagus salvinia, right? This is belongs to the Lepidopteran. To again, they introduced in US, Oregon State, right? Oregon State is Tyria, right? Tyria species use this tansy wort. We will say beetles also they introduce nowadays. These tansy also. This is these two. Right. Another one is another important. It's the first classical biological control agent introduced into Australia. It is very successful to control the cactus in everywhere because it's a dry continent in Australia. Some parts, so the cactus was a very big problem. But they have done is they have identified some of these the cocchinal insects, right? Cocchinal like a mealybug-like insect. They introduced this one from India to Australia. It was very successfully. They have controlled the cactus. So, right, see how these insects, they are making and eating. Right? It's look like a mealybug, right? But its color is different. So, this is Dactylophidae, right? Dactylophidae family. The species is Dactylophilus, right? This is Dactylophilus, right? So, Opuntia species and diphtheria, right? Another one is a diphtheria, Tephtidae. Right, they introduce. So these what they have been doing is like in oviposition time they are making holes. Through these holes, so secondary insects can go and it can kill the weed species. Right, this is they are making galls also. Right, they are making galls also. So another one is they use the alligator weed to control the thrips. Alligator weed control, they have used some of the thrips also, but introducing these ones are dangerous. If it is host range is high, it can target the main crop. Another one is a mite, right? Titanichus species they use in New Zealand, right? Titanichus in New Zealand to control these ghost weeds, right? To control the ghost weed in New Zealand, they introduce these ghost spider mite. We call it as a ghost spider mites belong to Titanichus species. The areophid mites they also introduced to control the gall forming, right? So this is if it is a mite attack, it will form the gall. It will gall form skeleton weeds, right? This was we call it as a skeleton weed to control these. These are the successful biological control. I put these pictures and names because these are successful biological control agents of the weeds in different part of the world. Right, so these are the end of the weed control agent, how you can use weed. So I haven't discussed the another important concept of the Padinium management. So in Sri Lanka, so I wrote an article, whether you read or not, I don't know, in the paper. So this is one of the biological control agent belongs to Coleoptera. We call it as a zygogram. This psychogramma is now available in Sri Lanka to manage the Padi. It was not successful, but in northern part of Sri Lanka, the first time of the history, I identified that one, and I reported even in this there in the Padinian newsletter. You can read, right? So it may be spreaded, overspreaded. It come across from one country to another, from India to Sri Lanka, because it's very closer to the north region. It is came in zygogramma. It is controlling pathogen, but the problem is farmers spraying chemicals, broad spectrum. So that's what it is difficult to manage this zygogramma population. Otherwise, it is working, right? So now we are moving to the last part of the section, is biological control of the plant pathogens. Until now, you won't ask any question. Please, you can ask. So biological control of the plant pathogens, right? So plant pathogen. So this biological control of the plant pathogen is an important, because it's a plant protection course. So you would have studied in the 2-2, right? That's how second year, second semester in the phytopathology course also, biological control of the plant pathogen. Now I am just touching, right? So these pathogens also we have to study, because these pathogen we have to control. The plant protection course is covering both pests and diseases. So biological control of the plant pathogens means, so plant pathogens, if you are controlling using the another microbes, it is very effective and it is not detrimental to the human being, because if you are using the chemical fungicide, these are nasty, right? So the biological control agents, they are having the three broad mechanism. One is an antibiosis. So an antibiosis is one organism, the inhibition of the one organism by another organism by producing some secondary metabolites. These secondary metabolites we call it as an antibiotics, right? So these antibiotics, if it is produced, other organism cannot grow, because this is toxic. This we consider as an toxic to the other, right? So this is mainly many fungal groups. Right? From that one, we are using the tetracycline. So antibiotics we are enjoying, right? Enjoying. But in case of this plant case, we can use this Bacillus cereus, right? Sweetmycin. So the example is a sweetamycin. It's an antibiotic produced by the Bacillus cereus. So bacteria are rarely producing antibiotics, but this Bacillus cereus is producing the sweetamycin, is first identified in Phytophthora rot in alfalfa, right? And also trichodoma also produces, right? You all know the trichodoma also produces many antibiotic secondary metabolites chemicals. And also we just last year in our research, we have identified there are 3 fungi we were testing on the again extraction of the Phytophthora infestant secondary metabolites against wheat, right? And of the gray mold fungi extraction and Fusarium solani extraction against wheat, it was working, right? It was just, it's another mode of the action. Nowadays, the biological control agent people, they have reported that this is induce the immunity of the plant. It's a mode of attack of it's a way of biological control of pathogen, but we are not directly using the pathogens or biocontrol agents. We are extracting the secondary metabolites of the plant pathogen, the same, the pathogen that are causing the diseases. We are spraying that secondary metabolites. Therefore, the secondary metabolites induce the immune. Because why? The plant will think that this is pathogen. It will start to react against that pathogen, the pathogenic substances. But pathogen will successful if it is penetrated only. Secondary metabolites cannot work without penetration. Right? That's how they are spraying. Now extraction and spraying in this way. So we are trying to induce our immunity. What we are doing then vaccination for us. Right? Very delicate pathogen. We are introducing out of blood no, to create the like immune. So that's how we are using. Right? That's how also we are using. And another one is a nutrient competition. So this biological control agent will grow very quickly and draw all the nutrition and will not allow the pathogen to get in touch with this carbon, nitrogen, oxygen, iron, whatever they needed for their growth. If there is no food, then pathogen will not grow. That is why normally we are using this Pseudomonas fluorescens, right? Normally Pseudomonas fluorescens you can find around the rhizosphere, right? Around the rhizosphere, they are quickly grow and cover all the nutrients. That mycorrhiza, another one is there. So quickly cover the root zones. So there is no food or habitat for this pathogen to grow, right? So this common way of organism limit the growth. Their target is limit the growth. So Pseudomonas fluorescens, they have been easily used to manage these bacterial blotches, right? Bacterial, so blotches. And destructive mycoparasitism. Destructive mycoparasitism means so parasitism of one fungus by the another. This is one habit of Trichoderma too, right? Other than the secondary metabolites, Trichoderma what will do is it will grow over the pathogen, other pathogen, and produce penetration peck over the other pathogen, and penetrate and reproduce into the other pathogen by direct contact, producing cell wall degrading enzyme. Some produce antibiotics. So in this way, they are destructive. Destructing is different from antagonism. Antagonism is a suppression. Destruction is direct killing, right? Direct killing by attacking, right? So this is important. It is importantly noted in Trichoderma. Trichoderma hasian. This Trichoderma can grow on nematode, eggs, and juveniles, right? Eggs and juveniles, it has been reported. So destructive. There are 3 more than 4 more. Earlier you were discussing the 3 mode. One is antibiosis, nutrition competition, and destructive mycoparasitism. Another one nowadays they are induce immunity by using the secondary metabolites. Okay. So the requirement of the biological control agent of the plant pathogen have effectively biocontrol agents must be obtained and produced, right? You have to get it. If it is very difficult to isolate from somewhere else, and if it is very difficult to mass multiply it, it is no meaning, right? So, and also it should be able to compete and persist. Should be able to compete with the pathogen. It should always persist in the land. No need to apply for continuously. In that way, this behavior should be the highly effective biological control, and to be able to colonize and proliferate. If you are introducing any environment, it should colonize and proliferate. It should be a non-pathogenic to the host and environment. In any condition, it should not be a pathogenic to the host. And inexpensive the production and formulation against, right? So it should be an ineffective. So for example, in the trichoderma, you can grow anywhere, any substances you have, or extracted coconut traps, whatever the ground grains, anywhere in any condition, trichoderma you can mass cultivate. Right, in our department, so nowadays we are producing a mass cultivation of trichoderma and we are selling to the farmers, right, to control many root diseases. The production must be result of the biomass of the excellent self life. In any biomasses, it should live excellent. If it is growing in the waste material like trichoderma, it is very easy, right? It is very easy. And delivery application must permit full expression of the agents, right? It indicates that if you are delivering in somewhere else or while transporting, it should not lose its virulence, right? It should not lose it. Virulence must ensure the agent will grow and achieve their purpose. In every way, if it is inoculated, it should grow and multiply and achieve the purpose. Otherwise, it is not considered as a successful bioagents. One such example is an trichoderma. So trichoderma in all agricultural soils, we call it as an plant doctor fungi. It has met all the qualities of what are the characters and specific characters a biological control agent should have. This is met by this trichoderma only. That's how everywhere we want to use it, we can use this trichoderma very successful. Even the Pseudomonas fluorescens are very difficult sometimes, but trichodermas are very successful and popular. And we call it as a plant doctor fungi, mean any type of soil-borne pathogen, it can work, right? And it can work. So the resistant is these many strains has been reported. Trichoderma viride, Trichoderma hasianum, Trichoderma hovaski. So different strains are available and commercially used, right? For the different fungus, any types, right? So applications and other things would have learned in the maybe second year, second semester. You can use this one as a seed treatment and foliar application, or you can mix to with any compost material you can apply. Right, in any condition, any way you can apply it. But if you are applying that, that field should be free from fungicides. If you are giving the fungicides, then it definitely it will kill the trichodome. Not only the trichodoma biological control agent, for the what other remediations also trichodoma they are using, especially removal of heavy metals and removal of toxic material from these aquatic region. Nowadays the experiments are going on. This is a picture showing how trichodoma growing over the pythium, right? Pythium, and it's attacking. Right? Again, so pathogenic fungi, this bacteria, the trichodoma, how it is working. So they are producing these enzymes, pectinases, proteases, and glucanases. You see, they are making damages of the cell. This is an electron microscopic picture. It's an electron microscopic picture. Some bacteria, so the trichodoma also can penetrate root cells. Through the roots also they can penetrate and it can attack. Nowadays, so commercially available products are there T22, trichodoma 22 against Pythium rhizoctonia fusarium. Right? Another one is, so the future developments of the trichodoma is needed sometimes. Why? Because whichever the gene responsible for the pathogenicity. So if you are inducing that pathogenicity gene to become more vigorous, then it will work very efficiently. That's the way the future researchers are needed. Right? Future gene modifications, right? Gene modification or silencing of unwanted deleterious gene from this. In this way, the future generations, future development should happen. Right, this is an end of this pathogenic control by using the microorganism, plant pathogens, right? So the overall for the biological control agents, introduction, multiplication, monitoring, who are responsible? First is the State Department of Agriculture. So if you want to introduce any biological control agent into the Sri Lanka, so Department of Agriculture is responsible. Under the Ministry of Agriculture, they have given the permission to the Department of Agriculture. So DG approved one only. So the Minister of Agriculture is have to sign it. Then only you can introduce any biological control agent. Even if you have introduced any biological control agent introduced into the country, then who are the people who have the authority to mass multiply and sell it? Right? Mass multiply and sell it. So, right, so they are the people coordinating the biological control of effort. Other one is the state universities and the research organization. And the Department of Agriculture, Extension and Research Offices are there. And the state universities like Faculty of Agriculture. So they can identify biocontrol agent. So they can introduce biological control agent with the permission of the Ministry of Agriculture for the research purposes. When it is successes, they have to get the approval from the ministry. Then only they can mass multiply and sell it or hand out to the Department of Agriculture to take the further process. Right? So they are the people to have to educate the public. The Department of Agriculture Extension can do that, or the universities. Universities have to do the extension education program through the extension department. So nowadays, so the Department of Agriculture Economics role is to develop an extension program. So they have to coordinate these education program to the publics, right? The publics. And the professional organization who are responsible. Nowadays, many companies are there. These companies can able to mass multiply and import that one with the permission of the Department of DG Agriculture and the Minister of Agriculture to the country. And it is approved only, they can mass multiply and sell it. So any professional organization, they are registered to produce biocontrol agent for testing and researches. They are who are responsible for increase the biological control and boost the biological control program. And private industries. Some of the private industries mean farmers also can do that one. Farmers can do that one. So funding agencies, some of the funding agencies can also do that. Farmers and growers, who are the people responsible for that, increase the biological control and the application of biological control agent and practically feasible biological control program throughout the country to minimize the chemical control. So if this is the end of this presentation of the class, and if you won't ask any questions, so you can ask any questions if you want.